Hello, my name is Khaled Jumah, and I am a bioinformatician at the University of Bradford. This poster represents our contribution to the Elixir Human Copy Number Variance project, where we worked on implementing copy number variance detection and federated sharing into the Galaxy platform. Galaxy is a web-based platform that provides tools, tutorials, and computational resources, allowing users to run complex analysis on their data without needing to install anything locally. Copy number variants, or CNVs, are large deletions or duplications in the genome that can impact gene function and are linked to various diseases, including cancer and rare disorders. Beacon version 2 is a global alliance protocol designed to enable secure, federated querying of genomic variants across datasets using a standardized API, making it easier to share data while protecting privacy. We focused on building a copy number variance detection and beacon integration workflow inside Galaxy. To do this, we implemented a suite of tools. CNV Kit as the main tool for automated copy number variance detection. The CNV Kit is a toolkit with many tools. One of them is the CNV Kit batch, where you can run the whole pipeline to detect the CNV kit, the CNVs, or if you want a more thorough query, you can use the rest of the tools that are available on the toolkit. All of them are wrapped into Galaxy and can be used in the Galaxy Europe or from Galaxy France or Galaxy Australia. You have then the CNV VCF to JSON to convert the CNV kit generated VCF file into Beacon version 2 compatible JSON4 file. Beacon 2 import and Beacon 2 search to load and query the CNVs within the Beacon system. All tools were integrated into Galaxy and published on the Galaxy toolshed for community use and are also available to install through pip or conda. We also developed training materials as part of the Galaxy training network, including a tutorial for querying the Beacon using version 2 queries, a step-by-step guide on how to build and deploy Beacon version 2 server using our tools. Also, it includes the Beacon 2 RI implementation study tools on which require an advanced knowledge on how to use terminals and Docker containers. You can find here also the workflow that we have developed to detect copy number variants on Galaxy using the CNV kit. The datasets are obtained from EGA rare disease synthetic data. The pipeline starts with the SAMTools view and reheader to adjust chromosome naming. Then it uses the BAM left align and SAMTools CAMD for cleanup. Then CNV kit batch to detect the copy number variants. This will generate a CNS file format, also called the copy number, the CNV calls. This will be used by the CNV kit export VCF, which converts the CNV calls into a VCF format. And finally, the CNV VCF to JSON to generate a Beacon compatible JSON file. To interact with Beacon database from Galaxy, we developed two tools, the Beacon 2 import and Beacon search. And as I have said, both of them interact with the Beacon database protocol directly from Galaxy, and it shows the research query from Galaxy or upload the data from Galaxy. The Beacon import, both of them, Beacon import and Beacon search, will check the authentication that the user who want to access the Beacon database has. If they have the permission to upload data into the Beacon database, it will check if the user wants to remodify the collections on the database by give him the question or the ability to delete the collection before uploading the data. This to avoid any duplication. The beacon search will check the authentication of the users by seeing what kind of permission they have. Do they have the permission to get the whole record? Or they got the permission to check if the record exists or not? If they have the permission to get the record, it will perform two types of main queries. One, if they're looking for the genetic variants, which are the data collection. These include the structure variants or the copy number variants, and they can use different parameters in the search. So if they're looking for a bracket query where they're looking for the variants in a specific location, or if they're looking for a copy number variants, or they're looking for a variance in a specific gene. They also can search the collection, the metadata collections, if they're looking for records for the metadata or for the study. So if they're looking for a specific analysis performed, or if they're looking for a specific cohorts, disease, or any kind of the metadata connected information from the GA4GH standard. To summarize, the integration of CNV kit and Beacon version 2 in Galaxy empowers clinicians and researchers to use Galaxy infrastructure to detect copy number variants and also to share their results with the other colleagues in a federated and secure method. Thank you everyone for listening.